My name is Alex Parker. I'm an assistant professor at the University of Montreal in the Department of Neuroscience. I've been studying ALS for about 5 years now. The past few years there's been an explosion in the genetics of ALS, and that's what drew me to it, because I thought perhaps there was something we could do here, apply our approach to have a better understanding of the disease. What we're trying to do is find new therapeutic strategies for ALS. Some of the recent genetic advances have identified many genes that are involved in ALS, but we still don't really know what these genes are doing under normal conditions and what's happening in the disease state. And it's hard to design a therapy unless you really know what's going on. So we have an alternate approach that we hope will be successful. Many research labs across the planet use models to study things, aspects of biology, as well to model human disease. There are things like computer models, cell culture models, and there are animal models. Most people are familiar with the mouse as a model to study biology, as well as to study human disease. What we use is another simple animal model. It's a worm. It's called C. elegans. C. elegans is a small worm. It's about 1 millimeter in length. It's not harmful to humans. There are parasitic worms, but this is not one of them. If you went outside, you could find it in the soil. The bonus to the system is that the animals don't live very long. They live about 3 weeks on average. So you can conduct experiments in a short amount of time. But how do we do this? Use C. elegans for ALS. So C. elegans was originally chosen for its neuroscience applications. So the worms have a nervous system, but it's much simplified. For example, it's estimated that humans have on average 100 billion neurons. Worms have about 300, so it's much simpler. But within that nervous system, there are motor neurons. And as you know, motor neurons are what are affected in ALS. So we're wondering if we could model ALS in the motor neurons of this very simple animal. So we took human disease genes that cause ALS, put them in the worms to drive expression of these genes in the worm's motor neurons. And what happens? Well, the worms get sick. It's not great for them, but it does give us a very powerful research tool. If you look at them, they start off fine, but they have problems moving and they ultimately become paralyzed. And if you look closer into their motor neurons, you can see the motor neurons are actually dying. All of this takes place relatively quickly, within 2 weeks. So this is a very fast system to study mechanisms of motor neuron degeneration. And as a geneticist, this is what you want. You want to have a phenotype. What we call a phenotype is some sort of observable difference that you can correlate with a genetic cause. Here, ALS genes in the worm's motor neurons causes the motor neurons to die quickly. And once you have this, we can then conduct additional experiments, what we call screens, to find ways to make these worms healthy again. So one example of our research is drug screening. We've used our C. elegans models to screen many thousands of compounds for drugs that would make the worms healthy again. And we found a number of compounds that work very well to reduce motor neuron degeneration. But what's next? Having a healthy worm is one thing, but as useful as they are in the lab, they're still very far away from humans, let alone ALS patients. So we're very fortunate to have colleagues, excellent ALS researchers here in Montreal as well as across Canada, and we're working together as a team, and we're following one compound, and so far it seems to be working in multiple models beyond worms, including mice, and is the subject of a clinical trial at the moment. So what we think is that our model, C. elegans, is like a scout system that allows us to find mechanisms and drugs quickly and relatively inexpensively, because it's much more difficult to do this in more advanced models. But the real strength of this is us working together to move things from simple systems closer to a clinical setting. None of this would have been possible without the support of ALS Canada and the donors who support ALS research. As somebody who was new to the ALS field, the Bernice Ramsey Award that I won just when I was starting my lab made all the difference. It gave us the means and the opportunity to make these models, and I don't think it would have been possible otherwise. Since then, our lab has doubled in size, our research is advancing, but we're far from done. We still need to find new mechanisms and better ways to treat ALS. And we're very hopeful that what we're finding in the lab will one day lead to treatments for ALS. So thank you.